Welcome, my dear students. Today again we are going to discuss some new topics of chapter number 5, that is the fundamental unit of life. In previous session we studied about history of cell, in which we studied about cell theory and the contribution of various scientists for increasing the information about cell and shape and size of the cells, structural organization of cell, and we also learnt about plasma membrane and osmosis and diffusion process. In today's session, we are going to study about some more characteristic features of plasma membrane, cell wall, nucleus, and cytoplasm. So let us begin. Water crosses the cell membrane in both the directions. But when more water leaves the cell becomes shrink, and this condition is known as plasmolysis. When the cell is shrink because of loss of water. When a cell is kept in a hypertonic solution, that time also the same thing happens. That is, because of the high concentration outside, the water moves from the cell to the outside environment. As a result of this, the cell shrinks, and this condition is known as plasmolysis or plasmolyzed condition of cell. In this slide, you can see the structure of plasma membrane, in which you can see the red color lipid layers, two lipid layers, and you can also see that the plasma membrane is able to transport oxygen, carbon dioxide, and water molecule across the membrane in both the directions. So these directions are showing you that this transport takes place in both the direction, and plasma membrane helps in transportation of oxygen, carbon dioxide, and water, etc. Osmosis is a special case of diffusion which takes place through permeable membrane. Importance of osmosis is great for unicellular freshwater organisms as well as for plant cells. Plants absorb water and nutrients from the soil through the process of osmosis. So here in this slide, you can see diffusion and osmosis in unicellular organism, where the picture of unicellular organism paramecium is taken, and you can see that oxygen is coming into the cell of paramecium and carbon dioxide is leaving paramecium cell. So you can see that transportation of various gases also takes place in unicellular organism through diffusion and osmosis process. The importance of diffusion is exchange of gases and water in cells. Cells obtains nutrition from its environment by diffusion process, and it helps in transportation of various molecules across the plasma membrane. And plasma membrane is a flexible and is made up of proteins and lipid. It can be seen with electron microscope only. The flexibility of plasma membrane enables the cell to engulf food, and this makes a special feature in living organism and a special process which is known as exocytosis and endocytosis. Exocytosis is a process in which material inside a cell is packed into a vesicle, and then this packed vesicle is excreted out of the cell, and this process is known as exocytosis. Endocytosis is the opposite process, in which the plasma membrane invaginates or folds in such a way that it makes a circular vesicle. It makes a packaging structure which is known as vesicle, and it folds inward to form a vesicle and it brings the material from outside environment into the cell. Now this will be more clear to you through this slide. Here you can see that in exocytosis process, the invagination is made in this internal membrane. This is the invagination, which is folding of plasma membrane or the internal layer of plasma membrane. Then after this invagination, this invagination detaches from the internal membrane and forms this vesicle. In this vesicle, the waste material is packed inside it. And after some time, you can see that this vesicle moves towards the outer periphery of the plasma membrane, and it ultimately attaches to the plasma membrane, and then it releases the waste material out of the cell. So this is the outer environment in which this waste is thrown out. So this process is known as exocytosis. Opposite to this, the same thing can happen in different direction. That is, if suppose this is a food material for a living organism, it has to take this food material from the outer environment. Then this will first come near to the plasma membrane. Plasma membrane will invaginate or make the folding and will enclose this food material in such a way that it will form the vesicle here. You can see the vesicle here. Now this food material which was earlier outside the cell is a part of a cell. So this process is known as endocytosis. Now we will talk about cell wall. Plant cells have outer rigid covering made up of cellulose which lies outside the plasma membrane. Cellulose is a complex substance which provides structural strength to the plant. In this slide, you can see the complexity of the structure of cell wall. Here you can see that cellulose, microfibrils are there, pectin is there, hemicellulose is there, and there is a very compact structure of cell wall. So chemically, cellulose is a complex structure. That is why it provides strength and rigidity to the cell wall. Here again in this slide you can see that what happens to a cell when it is kept in hypertonic, hypotonic and high isotonic solution. We have already done this particular part in our previous session, so we need not to focus on this again and again. Just go through the picture. These pictures are very important. Through that picture only you can understand that what happens to the cell when it is kept in three different types of solutions. Now let us again focus on to the cell wall. Cell wall is specifically present in plant cell and it is made up of cellulose. Cell wall permits the cell of plants, fungi and bacteria to withstand very dilute hypotonic external media without bursting. The cell tends to take up water by the process of osmosis. The cell swells, building up a pressure against the cell wall. The wall also exerts equal pressure against the swollen cell. Because of their walls, such cells can withstand much greater changes in the surrounding medium than animal cells. According to the chemical composition of different regions of cells, different region of cell gets different colors when they are stained. Children, let us go back to the first lecture when we were discussing about visualizing a slide under compound microscope. That time I told you that if you want your visualization very clear, we need to stain the slide. Actually, it is not a staining of slide, it is a staining of a sample. So what is exactly that staining? Why that staining is required? What are the things with which that staining is done? What is the importance of a staining? There are so many questions which can arise in your mind. So staining is a very important technique which makes your observation of any sample very easy. Children, when something is colored, you observe that thing very easily. The observation, the visualization becomes very easy when something is colorful. In biology, we have different types of chemicals which have different color and which particularly impart a color to a specific part of a cell. Like for example, if you want to visualize nucleus, we will have different types of stain which will specifically go and attach or bind to the nucleus part and will highlight the nucleus into the cell. So when you will observe a cell under a compound microscope, then that time your observation will be very clear. It will be very easy for you to just focus your attention towards the nucleus and to study the structural composition or to study other things related to nucleus. Similarly, we have different kinds of stains in biology which specifically bind to some different or some particular kind of organelle in a cell. Some regions appear darker than other regions in the cell. So, For this purpose, we have various stains. These stains can be iodine solution. It could be safranin, which gives a pink color to the cell sample. It could be methylene blue also. These three are the major stains which are generally available in most of the laboratories. That is iodine solution, safranin stain, and methylene blue. And staining is a technique which helps us to observe this slight sample under a compound microscope. It imparts a color to your material to which you are going to visualize in compound microscope. When the material gets stained, get colored, your visualization becomes very easier. Now let us focus on to another important, I would like to say most important organelle of a cell, that is nucleus. Here I have mentioned that it can be considered as the brain of a cell. Why brain of a cell? Because it controls the activity of all other organelles of the cell. So nucleus is the major part of a cell because it helps in cell division, it helps in formation of new cells, and it also controls the activity of other organelles which are present in the cell. So look at this slide, you can see that the structure is quite complicated and it is very interesting. It clearly shows that there is a nuclear envelope, means this nucleus has some outer layer. You can see here, this is a nuclear envelope. And this nuclear envelope is made up of two membranes, outer nuclear membrane and inner nuclear membrane. And inside this nucleus, you can see a thread-like structure is present. These are your thread-like structures. See, these structures are known as chromatin fibers. Chromatin fibers. And in this structure only you can see that there is a central part which is very highly demarcated here. So this particular part which is highly demarcated here is nucleolus. Sometimes children get confused between nucleus and nucleolus. So nucleolus is a part of nucleus. So this highly demarcated part is nucleolus, and this is the complex structure of nucleus. So let us begin with the nucleus structure, that is having two nuclear membrane. This is the inner part of this nuclear membrane is inner nuclear membrane, this one, and the outer part of the membrane is known as outer nuclear membrane. And you can see this nuclear membrane is not a continuous layer, it is broken. See, so the part where it is broken is making a channel or a entry for other substances to come inside the nucleus and also go outside the nucleus. So these are making some pore-like structure. These pores are known as nucleopores. Inside the nucleus, there is some liquid which is known as nucleoplasm. And again, here chromatin fibers are present and this particular structure is nucleolus. So this is about the anatomy of nucleus. The nucleus has a double layered covering called as nuclear membrane. It has pores to allow the transfer of various material in and out of the nucleus. The nucleus contains chromosomes which are visible as rod-shaped structure only when the cell is about to divide. When the cell is doing preparations for cell division, that time only these chromosomes are visible to us. These chromosomes are very important part of nucleus. These chromosomes contain information for inheritance of characters from parents to the next generation in the form of DNA. Chromosomes are made up of DNAs and protein. DNA is deoxyribonucleic acid and proteins are histone proteins. DNA molecules contain the information which is necessary for constructing and organizing the cell. The functional segment of DNA are called genes. Again, see, there is one more minute bifurcation is there. That is, first of all, you will have nucleus. In that nucleus, you will get chromosomes. Chromosomes are made up of proteins and DNA. The functional part of that DNA is called as genes. The DNA contain so much of non-functional part and a very small functional part. The functional part which is present in that DNA is known as genes. It has genes present in it. The cell which is not dividing, this DNA is present as a part of chromatin material. Chromatin material is visible as entangled mass of thread-like structures. Whenever the cell is about to divide, the chromatin material gets organized into chromosomes. Means when the cell is not dividing, when the cell is in the resting phase, that time it looks like an entangled mass of some fibers. But when the cell starts preparation for cell division, that time these entangled mass of fibers arrange themselves in a very specific pattern, and that time only these chromosomes are visible to us. They get organized for further division in them. The nucleus plays a central role in cellular reproduction, in which a single cell divides and forms two new cells. Along with the environment, it helps in determining the way the cell will develop and what form it will exhibit at maturity by directing the chemical activities of the cell. Children, this nucleus is very, very important part of the cell because it helps in cellular reproduction. In some organisms like bacteria, the nuclear region of the cell may be poorly defined due to the absence of nuclear membrane. That means in that bacteria, it may be possible that nuclear membrane will not be present in them, or it will be poorly defined. Such an unidentified nuclear region containing the nucleic acid, which can be RNA or DNA, is called nucleoid. So again, in bacteria also this DNA or genetic material is present, but that genetic material is not packed in a nuclear membrane. It may be freely floating in the cell. It may be unorganized, undefined region, and that undefined region is known as nucleoid. Such organisms whose cells lack the nuclear membrane are called prokaryotes. Children, it is very important to note here that what are the differences between the nucleus of prokaryotes and eukaryotes. We are eukaryotes. Our nucleus is defined in a nuclear membrane. It is very well packed and arranged. Whereas in case of some of the bacteria and other prokaryote organism, the nuclear region or the genetic material is not properly packed in any membrane. It is not organized, it is not defined. So such type of genetic material is known as nucleoid, and it is present in prokaryotes. Pro means primitive or primary. Karyote means carry on nucleus. So that was the primitive nucleus which was not organized. Prokaryote organisms were the primitive organisms in which the genetic material was not organized or it was not packed in nuclear membrane. But in them, in place of that nuclear membrane, nucleoid was present. Organisms with cells which are having nuclear membrane, like us, are called as eukaryotes, which are the advanced form of karyons or nucleus, in which the nucleus is packed into the double membrane layer, outer membrane and inner membrane. So here you can see the picture of prokaryotic cell, or you can say that it is a bacterial cell. In this bacterial cell, you can see this particular part is some highly demarcated part. So this part is nucleoid. In this nucleoid part, you will see that there is no nuclear membrane. It is not packed in any membrane. And in this bacteria, you can also see some ribosomes are there, which helps this bacteria to produce its own protein. For protein synthesis, these ribosomes help. And then you can see plasma membrane is also there, and cell wall is also present in these bacteria. And then you can see big flagellas and cilias around the body of this bacterium. These are the flagellas, and these are small hair-like projections which are known as cilias. So this is the structure of prokaryotic cell. Children, you need to draw this diagram into your whatever book in which you are maintaining your record. You need to draw this diagram there. Prokaryotic cells also lack most of the other cytoplasmic organelles which are generally present in eukaryotic cells. Like for example, when you will study about the eukaryotic cell, you will see that Golgi complex is present in them, endoplasmic reticulum is present, lysosome is present, and so many membrane-bound organelles are present in eukaryotic cells. All these membrane-bound organelles are absent in prokaryotic cells. Many of the functions of such organelles are also performed by poorly organized parts of cytoplasm. The chlorophyll in photosynthetic prokaryotic bacteria is associated with membranous vesicle but not with plastids as in eukaryotic cells. Now let us focus on to the other important part of the cell, that is cytoplasm. Children, this word can be broken into two parts. Cyto, that means cell, plasm means fluid. The fluid which is present in the cell is called as cytoplasm, which is generally not like the water. It has a thick solution which is major part of that solution is made up of water along with some salts, protein that fills the cells. This cytoplasm is surrounded by cell membrane. Nucleus of the cell is surrounded by cytoplasm. So here in the slide you can see that cytoplasm is shown here in this diagram. You can also see that nucleus is there, and you can also see other parts of the cell, nuclear membrane, cell membranes, ribosomes, etc. So cytoplasm is one of the very important part of the plant. So when we look at the temporary mount of onion peel as well as human cheek cells, we can see a large region of each cell enclosed by the plasma membrane. I told you in my previous sessions also that when you will visualize a cell under a compound microscope, you will see three main parts: cell membrane or plasma membrane, then you will see nucleus, and the third one is cytoplasm. The part which is between this nucleus and plasma membrane is cytoplasm. This region takes up a very little stain. When you will stain the cell with safranin, methylene blue or iodine solution, you will see that this particular region gets a very less stain. It is called the cytoplasm. The cytoplasm is the fluid content inside the plasma membrane. It also contains many specialized cell organelles into it. Each of these organelles performs a very specific function for the cell. Again, I want to remind you, I told in my previous classes that a cell also exhibits a division of labor because it has different organelles, and these different organelles have different specific functions within the cell. So the cytoplasm surrounds these cell organelles and provides it a media to perform its function. Cell organelles are enclosed by the membranes. In prokaryotes, beside the absence of defined nuclear region, the membrane-bound organelles are also absent. On the other hand, in eukaryotic cells, nuclear membrane is very well defined and is very well developed, and other membrane-bound organelles are also present in their cell. The significance of membranes can be illustrated with the example of viruses. Now we all are facing the problem of viruses, so as a human being we should have the curiosity to know about these viruses also. These viruses lack any membranes and hence do not show characteristics of life until they enter a living body or a living host. And once they enter into the body of host, they use the cell machinery of the host and multiply themselves into the host body.